with gave wonderful talks about using antibodies for treatment. And so some of this I'll mention again just to remind you some of the background, but I'd also like to make some extra points and focus on an aspect that I think we haven't discussed yet, which is the virus diversity of HIV and how you use antibodies in the context of the complexity of virus diversity. And so, as you know, for hundreds of years, it's been empirically known that you can give antibodies and they can help treat and prevent disease. However, recent technological advances have made it possible to make monoclonal antibodies. And so this slide is just to highlight how many monoclonal antibodies are currently being developed in phase 2 and 3 trials, specifically to treat infectious disease. And right now there are a lot of monoclonals that are more prevalently used for autoimmune diseases or in cancer. So why use antibodies for treatment? Why is this something that we should put any time into? So antibodies can have many effector functions, and some of these people have highlighted, and I'll just highlight it again. Using this slide that's actually from a review about cancer treatment. And monoclonals have been successfully used in the cancer setting, especially to treat B-cell lymphoma. And so this slide is a review about using the monoclonal rituximab to treat B-cell lymphoma. And so this is a monoclonal antibody that recognizes the CD20 molecule found on B cells. So it's the same thing as sort of giving a monoclonal antibody to target an infected cell, only in this scenario it's a tumor cell. And they're highlighting that through the constant region, many effector functions. So by targeting this cell, the antibody can mediate complement-mediated cytotoxicity to kill the cell. It can tag macrophages or phagocytic cells to come. This is called antibody-dependent phagocytosis, or ADCP. And it can also recruit NK cells to come and kill the cell, which is called antibody-dependent cellular cytotoxicity, or ADCC. And so just in general, it can tag this cell or opsonize it. And one mechanism that I wanted to add that's not included in this review, and Rick touched on this, and I've heard a couple of recent cancer talks, that they're beginning to appreciate the complexity, that oftentimes when you give a monoclonal, it is not one mechanism that is helping reduce tumor burden. It's not just ADCC, for example. And in fact, there have been some recent papers showing that there's incredible synergy when you use combination treatment, so including strategies that boost CD8s in combination with monoclonals are more effective in cancer treatment than using any single strategy by itself. And this is thought because when you tag a cell for opsonization with antibodies, it will increase presentation to dendritic cells, which will increase antigen presentation to CD8 cells. And so antibodies can serve as a bridge between different arms of the immune system. And this is just to highlight, as I'm sure you know, the antibodies sort of have two regions that we focus on. And this effector region that I was just talking about is found in the constant region, and this is the region that interacts with immune cells. But I'm more interested actually in the variable region, the antigen binding region of the antibody. And I feel like this is the real problem in HIV because having antibodies that recognize all the different kinds of viruses is a real problem because there's so much genetic diversity in HIV. And of course, as opposed to cancer in a virus infection, antibodies can also work simply by binding the virus and preventing infection, so aka neutralization. So just to focus now on how monoclonal antibodies can be used for HIV treatment or cure. This is sort of just a general list, and I don't really want to focus on specific treatments, but rather many of the speakers have mentioned different possibilities and strategies that people are using to achieve cure or remission, etc. And that really combination therapy is probably the best bet, where you don't want to use just one strategy or just focus on one arm of the immune system. You want to use combinations. And you probably also need to use combinations of antibodies. And so which begs the question, which antibodies do you want to use in any one of these strategies? And so that will be sort of the focus of my talk. And so first question, are all antibodies against HIV equal? Obviously not. And so this is a point that's been made before, but this is just another way of showing it, that it's been shown through years of research that the majority of people infected with HIV will start with one single founder virus. So this is just a simplification of genetic diversity with colors. And so if you start with one green virus, you develop antibodies that usually neutralize this green virus, but not other viruses. And so this person develops these antibodies, and they're called seroconverted. However, the virus, due to multiple reasons, high replication rates, large mutation rates, can quickly generate genetic diversity and essentially escape from this antibody response. And so you begin to have blue viruses, which are followed by blue targeting antibodies. And the green antibodies can't bind or neutralize the blue viruses, et cetera. And you go through years of replication in this cycle, and you end up with a person with lots of genetic diversity, virus diversity in their body, as well as antibody diversity. But these antibodies can probably only neutralize a small portion of each of the viruses in that person. And then I also wanted to take a minute, because we've been talking a lot about neutralization in the conference, to just talk about the methods that we use. And so the way we do neutralization assays commonly in the field is actually to split the HIV genome into two different plasmids. And so you have an HIV genome that is ONV deficient here, that you co-transfect with an envelope gene of interest. The envelope gene is the gene that will make the protein on the outside of the virion that is the target for antibodies. And so we call this pseudotyped virus, and you can swap in many different envelopes and have many different pseudoviruses expressing your ONV gene of interest. You then use this virus to infect target cells that express high levels of the receptor and co-receptor, and then these cells are made so that upon integration with HIV, they'll express the reporter gene luciferase, and then we can measure how much luciferase is present when you infect these cells with these pseudotyped viruses, and then we can measure how much that luciferase is reduced when we add in antibodies or we add in serum, and we can generate these neutralization curves. And you can measure shifts. So if you, here looking at neutralization of the black virus, say that's wild type, and then a different virus, which is green, you can see it's shifted to the right. It takes it takes more serum to neutralize this virus than it does this virus. And so you can sort of measure relative resistance or sensitivity by looking at the curve, and you can quantify it by extrapolating serum dilutions or concentrations that neutralize 50 or 80% of these viruses. So that's just to give you context for some of the numbers and concepts we throw at you. And so then really when John and Rick are talking at the VRC about broadly neutralizing antibodies, this is probably a representative panel from John's lab, but they have a really large library of different envelopes that represent genetic diversity found within HIV around the globe. And so you have an 174 ONV gene virus panel. This is the genetic diversity. This is a phylogram of all the subtypes that are represented within that panel. And so when something's broadly neutralizing, it's how much it's neutralizing all these different viruses. And so here is another paper. This was mentioned before, but this is another paper showing the same thing, how many people naturally develop this ability in their serum to neutralize these viruses. And so this is a paper more recently out of the Los Alamos laboratory, where they gathered data on 117 serum from chronic infection against 219 genetically diverse envelopes, and then graphed it. And you can see it's pretty linear association. So basically here they're graphing for each serum, how many viruses it neutralizes, the percent of viruses on this panel that it neutralizes, and how many serum neutralize that many viruses. So if you look at this dotted black line, and you look at how many neutralize half the viruses, it's about half the serum. And then if you define broad neutralization as 97% neutralizing this panel, only about 3% of serum can neutralize 97% of the viruses. And of course, because of technological advances, it's quite, and also knowing the structure of the virus, you can make much better virus proteins now, tag antigen-specific B cells, and pull out the antibodies that are responsible for this broad neutralization, and generate them as broadly neutralizing monoclonal antibodies. So not all antibodies are equal. Once you have these broadly neutralizing antibodies, are they all equal? No. So I wouldn't just swap in and out broadly neutralizing antibodies in any strategy you're going to use. And so you've seen this picture before. Antibodies, so this is a picture of the trimeric viral spike on the outside of the virus membrane, and it's coated in sugars or glycans. And broadly neutralizing antibodies, so one difference is they target different sites on the virus. And the extra point that I want to make is that for many of these antibodies, or for certain sites, the antibodies' epitopes incorporate glycan. So the antibodies not only see virus protein, but they also see the glycan sugar. And this is true for the V1, V2 antibodies that target the top of the virus. And this is true for the antibodies that come in the side and target the V3. And I think that's important for escape, is recognizing the role of glycosylation of the virus protein in recognition of the antibodies. And so again, you've probably already seen this, but I just want to highlight a couple things. So just by looking at these potency and breadth plots, so here, this is plotting at each dilution of antibody, how many viruses on those large panels it neutralizes. So if you just first look at, if you add the highest concentration of antibody, you can see really large differences in the ability of these antibodies to neutralize. So for example, 2G12 is not a great antibody. At the highest dilution, it neutralizes the fewest number of viruses. 10E8 is probably one of the best, so it's broadest. But if you start to look at concentrations, 10E8 actually needs a lot of antibody to neutralize this many viruses. It's not as potent. So as you use less and less of this antibody, it can't neutralize as many viruses, but then you look at an antibody like PGT121. Here, it does, at the highest concentration, it doesn't neutralize the most viruses, but it's very potent. And so as you use less and less of the antibody, you still maintain the ability to neutralize a lot of viruses. And so that's an important distinction that might be relevant when you're thinking about using broadly neutralizing antibodies for treatment. And the other point I want to make is that's just looking at the whole large panel. But within the panel, there are different subtypes, and antibodies can be better or worse against each subtype. So you can break up this data by genetic subtypes and see which antibodies are better against which subtype. It's a different way to look at the data. And so I just want to point out a couple of things. So you can see 10E8 here. So this is just showing, okay, so first of all, this is a much smaller panel. This is data that was compiled by the Los Alamos database. This is just on the internet of an 80 virus panel. And here I'm showing the percent of viruses neutralized by each antibody and the average potency for an IC80 of 10 micrograms per mil. And so the point just is that if you look at percentages, 10E8 can neutralize a large percent of almost all these viruses despite the genetic subtype, but it's not very potent. It's usually at over 1 microgram per mil. But if you look at an antibody like CAP256, VRC2625, it's not that broad. It's really bad against subtype B viruses. This is an antibody you probably don't want to use in a subtype B cohort. But it's extremely potent, especially against subtype C. So it's isolated from a subtype C infected individual, and it has incredibly low IC80 at 0.01 micrograms per mil. So this is just highlighting differences in antibodies, and then of course there's this history of many clinical trials where people have tried just infusing monoclonals on their own into people infected with HIV. And this was the previous studies with generations of antibodies that weren't as broad as potent as the new ones. And what they found basically is you can give a cocktail of these antibodies to people with viremia. You see a slight dip in virus load, virus comes back, or you can give it to people who are on antiretroviral treatment, take them off antiretroviral treatment in a treatment interruption strategy, and the virus comes back, but it's slightly delayed. And resistance develops to the antibodies, and you can document this. And then more recently, there have been trials out of the VRC and Michelle Newson's group at Rockefeller, also trying single antibodies that are sort of quote-unquote better, the ones we were talking about. And then this is just showing summary figures from all these papers. You kind of see the same thing where virus dips after you give the antibody, and then virus comes back. And you can document resistance in all these trials, that resistance is found in these viruses that are rebounding. So one question is, does pre-existing sensitivity to the antibody matter? Does it matter what kind of viruses you start these trials with? And it absolutely does. And so I'll just quickly go through some of this previously published data, and I'll focus on some of the work that I did in John's lab, analyzing the trial with VRC01. So what you can do basically is sequence the gene from each person enrolled in the trial at different time points. You can clone the envelope and then test it in neutralization assays. So here's just looking at the genetic diversity of 8 people enrolled in this trial. They're all from North America, the NIH. They're all subtype B infected, which you can see. And then you can also see that these people are chronically infected with HIV. And each person has their own identifiable branch of virus, but within that branch there's incredible diversity for each person, and that's indicated by the colors. You can clone these viruses and test their sensitivity. And what you can see is that just starting off, this is before they've seen any antibody, there are differences within that diversity in that person and how sensitive they are to the antibody that you're going to use to treat that person. And so you can see here in red and pink, these two people are starting off with pretty resistant virus to the antibody that they were infused with. And I guess I should link back, that these are the two people who are the most resistant to a drop in virus load. And so obviously it does matter, and it has functional consequences when you're looking at a decrease in plasma virus load. So that's for pre-existing diversity. Does de novo resistance develop after you treat someone with antibody? And here I'll highlight work from the Rockefeller lab where they treated people with 10-1074, which is an antibody that sees that V3 glycan site. So the glycan is actually very important within the epitope. And here are graphs they're showing of 6 people they infused with this antibody. The graphs are showing just the sequence for the glycan that's involved in this epitope. Wild type having the glycan there, which means the antibody can neutralize that virus, is quite common. It's quite conserved. And everyone starts off with gray wild type virus at 100%. So the y-axis is percent of mutant virus, and the y-axis is time after infusion. The antibody concentration is shown in white. So they infuse these people. They all have wild type glycan there. The viruses should be sensitive to this antibody. You infuse them with antibody. Over time, the antibody concentration decreases. Almost immediately, what you see that's indicated by color is that there's some sort of mutation in this glycan site. And when they sequence these people, what they could see is it took one nucleotide change that was non-silent to basically get rid of this glycan site, and the virus becomes totally resistant. And so it's really quite striking. And then as the antibody concentration decreases, some of these people go back to having wild-type virus, and some people don't. So the striking example of de novo resistance happening very quickly. Here for the VRC01 trial, I just wanted to highlight, so I'm just using this person as an example. This is their virus load in color. This is the antibody concentration in black on the same graph. You give the person the antibody, the virus load dips, and it comes back. And so we sequenced from this pre-infusion time point and then this post-infusion time point where you can see the virus load is completely back to set point, but the antibody is still there. So the question was, how is the virus getting back to set point in the presence of the antibody? And so what we did was clone a representative number of viruses from each of these people. And what we can see is that none of these people, except people who were fairly resistant to begin with, became completely resistant. For the most part, the resistance just increased slightly. You can really see it in 4 main people. So the viruses changed enough to become just enough resistant to the antibody to continue to grow in the presence of the antibody. And then we could look at the sequences and determine how did the viruses change. And what we saw was that there was no de novo resistance mutations, the way you saw from escape from 10-1074. What we saw was there was selection for more resistant strains, again, from the pre-existing virus diversity in that person. And so occasionally they had pre-existing unusual residues in the CD4 binding site. And a lot of people actually, what they did is that the V5 loop is part of this complex epitope of the CD4 binding site, and they could just change the loop either by making it shorter or longer. They could become slightly more resistant to this antibody. And then another point I wanted to make that's not in this paper is that we also took these same viruses. We could show, and this is just representative, we showed this in all the people, but this is just one participant. This person not only became more resistant to VRC01, but when you look at autologous antibodies, the person maintains resistance always to their own antibodies. And so you can look at that as well with these same viruses. So selection pressure is not only with the antibody you're infusing in, it's also with their own immune system that is maintained at this time. So it's also something you want to think about in cure strategies, that if you're counting on using autologous antibodies to participate, most likely all the viruses in that person are resistant to that antibody. On the other hand, viruses that are sensitive to the antibody, their own antibody, are not going to grow out. So after this, another question was, does plasma virus load matter? These are all people who are not suppressed. They have somewhat normal virus loads. they're treated with an antibody, you see a dip, the virus comes back. There's lots of replicating virus. What happens when you suppress people, give them an antibody, and then take away the antiretroviral treatment? Can the antibody on its own continue to suppress the virus? And so there have been at least 2 published trials, again from the VRC and the Nussensweig's group, using 2 different antibodies. And I'll just briefly go in showing that in both these trials, they sequenced, and it's 2 papers, but actually 3 trials. 2 trials with VRC01 and 1 trial with 3B and C117, which are 2 different broadly neutralizing antibodies. And you can go in and sequence virus and test resistance. And so here I'm showing from the University of Pennsylvania trial, they sequenced in these black rectangles viruses before this person went on antiretroviral treatment. So this is the chronically replicating virus diversity. And then they sequenced what rebounded in the presence of the antibody, and that's in yellow and red. And so what you can see is there's genetic restriction. So the antibody is exerting some selection pressure on the viruses that rebound. And there's a slight delay in how long it takes for virus to rebound. But virus rebounds, and it's slightly more resistant. So you can, again, clone these viruses and test neutralization and see that the selection pressure is obviously for viruses that are more resistant to the antibody. And then this is sequencing that I did from the NIH trial, again with VRC01. Here the colors are slightly different. Before art is in blue and rebound is in red. And you don't see as much genetic restriction in the people enrolled in this trial as the other ones. But the overall conclusion from all these trials is you can delay virus rebound slightly, the antibody exerts some effect, and viruses rebound that are mostly slightly more resistant to the antibody than the pre-existing viruses. So none of this is super surprising, but they did the clinical trials, and now we know exactly how this happens. And it's also important to point out that you don't need to be completely resistant to the antibody to grow. You just need to be resistant enough to be fit in that environment. So despite having high or low virus loads, no matter what you start with when you infuse someone with an antibody, you end up with a similar result. And so this has made me very interested in my own lab in how we evaluate pre-existing virus diversity in people and how that will affect antibodies that you pick for treatment. So how does virus diversity affect BNAb sensitivity? And these are studies that we started in GW in collaboration with Brad Jones's lab, and they're experts in analyzing virus reservoir. And so what I wanted to do was how to come up with a slightly more relevant model in how do you assess pre-existing virus diversity within someone who is suppressed on ARTs and test different broadly neutralizing antibodies to figure out which one is the quote-unquote best. And so for this project, we came up with a panel of 13 antibodies that are currently being developed for clinical trials or have been used in clinical trials, so we think there's some treatment relevance. And then I separated them here by the epitopes that they see. So we have the receptor binding site V3 glycan, V1, V2, and GP41. I'm not going to go through this slide. I think Brad will go through his methods, but I just wanted to make the point. So this is a collaboration with the Jones lab. These are methods that they use all the time. And the way that we were going to look at reservoir virus is that they receive leukapheresis PBMCs from different clinical cohorts. And the cohort we looked at here are all mainly people from Canada, so they're subtype B infected, and they have access to really good treatment. They're all suppressed on ARTs. They're leukapheresis. The Jones lab receives these samples, and they do these quantitative viral outgrowth assays that I'm sure Brad will go through. And sort of the point is, I just want to make a point that they do limiting dilution replicates of the CD4 T cells, and then they stimulate the virus to grow out. And so this is sort of more representative virus panel for us to look at as opposed to the large genetically diverse pseudovirus panel. And we know within each individual, we can have a couple different viruses from that person that's more representative of the latent virus in that person. And so they let virus grow out. They assess which wells are positive for virus and which wells are negative by doing P24 ELISAs. So this is a little cartoon schematic from a postdoc in the Jones lab. And so this is just representing, at different dilutions of cells, you have different numbers of positive and negative wells. And that they are giving us the supernatant from these wells for us to test in our neutralization assays. And so here's a graphs of all the data we can generate from one person. And so this is the patient or the participant from whom the Jones lab received leukapheresis samples. Here we tested 4 positive wells that I'm calling viruses 1 through 4. And then for each virus, we tested this panel of 13 broadly neutralizing antibodies. And we generate these neutralization curves. And then I'm separating them by epitope. So there are about 3 to 4 antibodies represented on each graph. And these are positive and negative controls. And then you can just sort of see by eye some of the data we can get. This person in general, all their viruses are neutralized by CD4 binding site antibodies. Excuse me. You see a little more diversity in the V3 glycan antibodies and V1, V2 antibodies. This virus is completely resistant. This virus is really well neutralized by all 3 antibodies. This virus is somewhat resistant to the V3. This virus is neutralized quite well. And then to summarize all this data, this is a really large heat map of all the IC80s. And so what I'm showing here is each participant is in a black box, and then each row is the viruses that we tested. So these are positive wells in the QVOA assay. And then again, each column is an antibody, how well it's neutralized, color-coded, with red being potently neutralized and green being less potently neutralized, white being resistant, organized by epitope. And so again, you can see some patterns. And here I'm summarizing numbers for percent of viruses neutralized, and then of the viruses neutralized, how potently are they neutralized here. And so you can just sort of see by color. In general, the CD4 binding site antibodies are pretty broad, but maybe not as potent. So you can see, for example, VRC01, one of the first antibodies to be pulled out, is not that great, actually, compared to some of the next generation antibodies. So VRC07 is a relative that's been optimized by the VRC, VRC07 523. And you can just look and see, yeah, it is broader and more potent, and so it's sort of a better antibody. VRC07, 3B and C, and N6 are all somewhat similar, actually, when you look at breadth and potency on this panel. Another striking thing is when you look at V3 glycan antibodies, they're less broad, but when they do neutralize, they neutralize better. And so you can see the difference here that it's mainly white and orange. That's sort of true of the V1, V2 antibodies. They're maybe a little less broad. You can see, again, this CAP256 antibody, which is really specific for subtype C viruses, does not do that well on subtype B viruses, except for these 2 viruses here, which is interesting, and I'll go back to that later. And GP41, so these really 10E8 antibodies are the best compared to 2F5 and 4E10. And this is an optimized 10E8 antibody from the VRC. And again, you can see that it's slightly more potent, and so it does perform better after optimization than the original antibody. This sort of falls into the category 2 of broad, but slightly less potent. Okay. So then we are interested in linking, and this is an ongoing project in our lab, this phenotypic profile with genetics of the virus. And so here I just wanted to delve into a little bit one of the most phenotypically different participants, and I had pointed out before this person with their graphs, they have different sensitivities to V1, V2 antibodies and V3 antibodies. And you can see that here, that half the viruses are sensitive to 10^74 and half are resistant, and half the viruses are neutralized by this PGDM 1400 antibody, and half of them are resistant, and it's the opposite too. So there's a lot of phenotypic diversity here. We wanted to get the source by looking at the genetics. And so here I'm just focusing on these bottom 2 viruses as representative and showing sequence alignments when we delve into the epitopes of these antibodies. So here I'm showing alignment of virus 4 and 5 within the V2 loop. And so what you can see is that virus 4 is neutralized by PGDM 1400, virus 5 is resistant. And what is known about this epitope in this antibody is that this glycan is very important for neutralization, glycan 160. The sequence for an N-linked glycan is N something serine or threonine. And what you can see here, so this is the glycosylation sequence right here. And what you can see is there's a difference in the threonine to alanine here. So that's the difference between having a glycan and not having a glycan. It's a single amino acid change that renders this virus sensitive to PGDM 1400 and this virus resistant. What you can also see that's sort of interesting is this antibody is really specific for subtype C, for some reason, really neutralizes these viruses. It's very rare in the panel that we were looking at. And this virus 4 is super sensitive to neutralization. Virus 5 is a little less sensitive to neutralization. And then what you can see going into the literature is that there's this lysine 169 that's very important for neutralization by this antibody. This lysine is very well conserved in subtype C viruses and rarely found in subtype B viruses. However, both these viruses have this lysine. So most likely the glycan plus this lysine is why this virus is very well neutralized by this antibody, but this is very rare. And then this virus has the lysine, but not the glycan, so it's neutralized, but a little less well. And then we can look at the V3 loop of these two viruses. And again, there's a glycan that's really important for neutralization by this antibody. The glycan sequence is right here, glycan 332. It's very well conserved. However, virus number 4 has a lysine in this position, abrogating this glycan, and this virus is resistant to 101074, and this virus has the glycan, and it's neutralized. So this person has a lot of unusual residues that give it this phenotypic diversity in these antibodies, or in the sensitivity of these antibodies. So how does this diversity affect treatment with monoclonal antibodies? So how do you sort of translate some of the in vitro assays we're doing into in vivo studies? And again, this is all sort of the start of projects, so none of these stories are totally finished. But I wanted to switch to focus on another participant, OM5001. It's been the focus of a collaboration of the Jones lab and Harris Goldstein's lab at Einstein College, and we've had the opportunity to do some virus sequencing with them. So this person has much less phenotypic diversity. We looked at 5 positive QVOA wells here, and you can see the patterns of neutralization of all the antibodies are pretty similar. And also I'd like to point out that all the viruses are pretty sensitive to 1074, few differences. So we started by sequencing just bulk the envelopes from these different wells, and what we can see is that, yeah, there's not a lot of phenotypic diversity, there's also not a lot of genetic diversity. There are 3 main branches within this person's virus, but they're not that different, actually, between the 3 branches. And this is maybe to be expected. This person was someone who was suppressed within 3 years of infection and has maintained suppression for 8 years. And so the Jones lab collaborated with Harris Goldstein's lab, and Harris Goldstein has a humanized mouse model. So here, just to show the brief schematic, the Goldstein lab has these immunodeficient mice that they can engraft with PBMC from people who are HIV infected but suppressed. And they're interested in looking at virus rebound when you engraft latently infected cells into these humanized mice. So what they do is intrasplenically inject leukapheresis PBMC from the Jones lab in combination with allogeneic irradiated PBMC that sort of stimulate an immune stimulatory environment to get virus, to get the CD4 cells activated and expressing virus. And then what we were interested in is actually sequencing the virus. And I was interested in, if you take the same leukapheresis samples and you put them in the QVOA assay, you put them in the humanized mouse model, what are the differences in virus diversity that grow out in each model? And so this was done with participant OM5001. And so here's data from the Goldstein lab, which is showing this person actually has a relatively high-ish reservoir. And so when he performed these assays in 12 mice, what you could see is that all 12 by week 1. So here's plasma virus load over time for individual mice in gray and the average in red. And that by week 1, all 12 mice are HIV positive, which is pretty unusual. And so here he's just quantifying the proportion of viremic to aviremic mice over time. And I just want to focus on this time point of week 1. Sorry, for some reason I did some weird animation here. But I just want to point out that this is the time point where we're sequencing. So I want to know what is the difference between this virus and 12 different mice versus the QVOA virus. So here I'm showing a maximum likelihood tree. We did SGA sequencing of 9 QVOA positive wells from the Jones lab and 5 positive mice from the Goldstein lab. And so the mice viral sequences, and this is whole full-length gene sequences, the mice are in green and the QVOA are in blue. And here's the tree. And what you can see is that they're pretty intermingled. That's the overall conclusion. However, then you can look a little bit closer, and you can see that there are 5 main clusters of viruses. Bootstraps are also shown, over 85%. You can see that there's this cluster 5, which is the dominant cluster, and it's the dominant sequence that we find in QVOA and in mice. So it seems like this person really has a lot of this particular virus. You can see a couple clusters that you only see in QVOA or you only see in mice. And I guess one question is, do you have to just do more deep sequencing, that we haven't reached the depth of sequencing, or that there really is a difference in how you stimulate latently infected cells into which viruses grow out? So you can see that this cluster is only found in one well of QVOA, and these 2 clusters are only found in 2 different mice. And so next, these collaborations allow us to test what happens in an in vivo model when you give an antibody to the virus diversity. And so again, these are experiments done in the Goldstein lab, where he took the immunodeficient mice, injected them with the same PBMC, so from the same participant, OM5001, and then this time gave an intravenous antibody injection of 101074. And then again, he looked at plasma virus load, and we looked at sequences of what kind of virus was growing. And so here are the same pictures from the Goldstein lab. And so one obvious thing, but here's the antibody concentration added. You inject the antibody, and these mice are suppressed. The virus is sensitive. We already showed that in the viruses from the QVOA. And so you can see that 100% of the mice are not viremic in the first 2 weeks. So there's a delay in rebound, which is again what you see in humans, even though this model is really not the same as humans. And so then here the question, we're sequencing the virus that rebounds, the first detectable time point at which you have a high enough virus that we can sequence. And so what you see here, actually, the other point I want to make is that virus rebounds when antibody concentration gets really low. So only when the antibodies out of the system does virus rebound. And so here I'm showing a slightly larger tree, where in purple are the sequences from mice treated with the antibody 101074. And what we see actually is genetic restriction. And so here you can see that there's this dominant clone that we saw in the mice and QVOA assays in the absence of antibody. And we actually only see 1 mouse whose rebound virus is this dominant clone. What we see is there are 3 other mice. 2 have outgrowth sequences that are found on this cluster that we've never seen before in these assays. And 2 mice have this other, less dominant cluster here. So an obvious conclusion would be that the antibodies are exerting selection, and that these viruses are resistant, and that's why they're growing out, and they're so different from what we saw before. And we haven't officially tested this, but I don't think that's true. And so one thing is, this rebound is only occurring in the absence of the antibody. And when we look within the epitope, all the viruses have glycan 332, which is so important for neutralization. So there's no obvious resistant markers in these viruses that are growing out. However, there's still genetic divergence, and it looks like some genetic selection when you are treating these mice with antibody. So that's sort of interesting, and we're curious about how that's working, and also we need to formally test resistance, because it's possible that resistance is just not found by removing the glycan for some reason. So these studies are ongoing, but in conclusion from all this work, what we can say is that a single antibody infusion in humans can decrease virus load and exert selection pressure on the virus quasi-species within that person. Virus populations that become more resistant to broadly neutralizing antibodies also maintain resistance to autologous antibodies, so it's important to remember that autologous immune pressure still remains even when you're treating with exogenous immunotherapy. Resistance can derive from pre-existing virus diversity or de novo mutations, and that's likely dependent on the antibody you're using and probably how complex the epitope is. It's probably pretty easy to get rid of a glycan and become resistant. it might be slightly more difficult to escape from a receptor binding site antibody with a single mutation. On the other hand, there's lots of pre-existing virus that's resistant to these antibodies, and we found from clinical trials that then that virus is the one that will become dominant. So really, I think a take-home message is that pre-existing virus diversity can really affect sensitivity to treatment with broadly neutralizing antibodies, and I think that this pre-existing virus diversity is a hard concept to replicate in animal models, and so I think it needs to be looked at in multiple ways, or taken into consideration when using animal models. And then finally, bNAb infusions into the humanized mouse model can exert selection pressure on rebound virus, likely in the absence of resistance. And so, I don't know, I'm intrigued by this observation, but it might be that the model is, artificial, it's a model. So I'd like to thank everyone that helped with these studies, so especially the clinical trials. There's always a lot of work, and this is a much reduced thank you and acknowledgement slide. But most important, the study participants who participate in these clinical trials where we really learn, about how to use broadly neutralizing antibodies. And then also all the participants from the Maple Leaf Clinic that collaborate with the Jones Lab. And then I'd like to thank the Jones and Goldstein Lab for these great opportunities to get samples from them. And Maria Coram in my lab, who performed most of the sequencing studies that I showed you in the second half of the talk. Thank you.
So, Rebecca, it's really interesting that people don't, if I understand it correctly, don't seem to often make antibodies that are, or viruses that are resistant to neutralization by glycan binding antibodies. Is that because these are our own sugars? And if so, these antibodies, those monoclonals that you found, were derived from some humans, and are there some humans who do this versus those, the majority who don't, and what characterizes those people who can make self-reactive glycan antibodies?
Oh, those are really great questions. Oh, okay. I think you sort of answered some of them yourself. So one thought is that people don't make a lot of glycan-only antibodies because that's a self-protein. And that's the difference with some of these broadly neutralizing antibodies. It's they have the ability to both see glycan and virus protein, maybe except for 2G12, but in general. Then I'd say it's also, there's a lot of literature supporting the fact that these broadly neutralizing antibodies are weird and break tolerant, and a lot of them are more autoimmune than your normal antibodies, and so that may also play a role. About the genetics of people who make broadly neutralizing antibodies that do target these protein glycan epitopes, I don't know if anyone's really looked at that. Or I don't know the answer off the top of my head in terms of the genetics. What I do know is that for whatever reason, when people study the development of broadly neutralizing antibodies against these sites, for whatever reason, there's some autologous antibody response against that epitope, and it might be that if the person's virus, that particular virus protein conformation is more immunogenic in that spot. So it might be some interplay between the genetics of the B cells in that person and the virus that they're infected with. But what you can see is that once you develop an autologous antibody response against that site, the virus starts to play around with different mutations in that site. And oftentimes, just in general, when you develop diversity within one of those epitopes, that's when you develop a broadly neutralizing antibody, because you start to get a lot of different antibody swarms targeting that site, and then there's an increased chance that the antibody is going to learn how to tolerate diversity in that site and be broadly neutralizing.
Actually, I have two quick questions. One is you have calculated IC80. Is this any specific reason to only calculate IC80, not IC50, not IC60? And the second, monoclonal antibody, like in the case of co-infection, like a person have like tuberculosis and HIV.
Oh, okay. Second question first. I don't really know how co-infection with TB affects the development of anti-HIV antibodies. Sorry, the first question, so you're asking about the differences in IC80 versus IC50. I would say, in my opinion, there's a lot of work recently. You know, every method you use to test something is slightly artificial. I would say that the IC80s and IC50s on the pseudovirus panel are relative measures of sensitivity. They're not exact. And so I think people choose to sort of show IC50 or IC80 depending on the point you're trying to make. So if you're looking at the difference of neutralizing a wild type or mutant virus, maybe you want to pick IC50 as that middle point on the curve is like the most accurate measure of differences between sensitivities and viruses. On the other hand, a lot of antibodies don't necessarily reach 80 or 100% neutralization. So sometimes when you're thinking about clinical trials and you really need the viruses to all be neutralized, you want to pick a higher bar, like an IC80. And again, that concentration is not the exact concentration that you should use in a clinical trial. It's more a relative measure that in this assay, an antibody with a lower IC80 is probably going to be a more potent neutralizer than an antibody with, a higher IC80. Is that?
Yes.
Answer?
Thank you.
On time. We have to finish the session here. Thank you very much. Thank you, Rebecca.
Thank you very much.